Hi, my name is Miss Deaver, and I want to welcome you to AP Biology. For today's lecture, we're kind of just going to go over a little bit of general review of biology before we really get into the course. So, AP Biology, what is this course? Well, throughout the course, you will study core scientific principles, theories, and processes that govern living organisms and biological systems. That being said, if biology to you seems really, really, really boring and you really want to be a business major, I hope you find this course exciting, but it might not be really fun for you. The course is broken down into science practices, skills, and content. So, what exactly are these science practices that the AP College Board deems necessary to learn? The science practices are skills that you are expected to develop and apply throughout the course. That way, you can apply these to the AP exam come May. These practices include concept explanation, analyze and visualize representations, determine scientific questions and methods, represent and describe data. This is pretty big on this one. I would say the first three FRQs in the test really need you to be able to represent and describe the data given to you. Perform statistical test and data analysis. Develop and justify scientific arguments using evidence. So we also have these things called the big ideas. The content covered in AP Bio is broken down into 4 big ideas. Big idea number 1 is that evolution, the process of evolution, drives the diversity and unity of life. We will hit um, evolution towards the end of this course. Energetics, the biological system uses energy and molecular building blocks to grow, reproduce, and maintain dynamic homeostasis. Big idea 3, information storage and transmission. Living systems store, retrieve, transmit, and respond to information essential to life processes. And number 4, system interactions. Biological systems interact, and these systems and the interactions exhibit complex properties. All right, so these big ideas serve as the foundation of this course and are further broken down into learning objectives. Define what a student needs to be able to do with the content knowledge in order to progress through this course. To progress through this course. Essential knowledge, describe the knowledge required to perform the learning objective. These are listed at the front of your notes for your reference. So if you look at the note packet that's been handed out in class, you'll notice every note packet is designed like this. The learning objective is always going to be on the left-hand side, as well as the essential knowledge. So the essential knowledge is really what you need to be asking yourself before you take the AP Bio exam. Can I answer these questions? If you can't answer any of those questions, the AP exam will be much harder for you. The course will cover 8 units, and yes, we will cover all 8 units. It's going to be a fast progressing class, but I believe that it's my duty to teach you all 8 units before you show up to the exam. Unit 1 is the chemistry of life, so this involves big ideas 2 and 4, and what you'll see through the theme of this slide is that all of the big ideas really tie into all of the units. Cells is big idea 1, 2, and 4. Unit 3 is cellular energetics. Unit 4 is cell communication and cell cycle, big ideas 2 and 3. Unit 5 is heredity, so that's going to be 1, 3, and 4. Unit 6 is gene expression and regulation. Unit 7 is natural selection. And Unit 8 is ecology. Notice, like I said in the beginning of the slide, the big ideas in each unit overlap and build on each other. What does that really mean? That means you cannot forget what you learned in one unit when you go to the next unit. because we will be testing on each of those units for every test. I'm going to say this now, and I hope you're paying attention. Every unit that you've learned previous to the test is up for grabs to be tested. This is a cumulative course. So if I'm testing you on unit 5, I have free will and ability to test you on 1, 2, 3, and 4. I believe that's extremely necessary because once you get to the AP exam, you will be expected to remember all 8 units. I graded questions 1 and 4 at the AP grading. Question 1, the first question was, what does a nucleotide compose of? That you will learn in this unit, Unit 1. The amount of people who showed up to that test for getting that information was shocking. So please remember, you have to always keep the information you learned throughout the year ready and available to recall for any unit test. Meaning, what you learn in each unit cannot be forgotten because it is important for other units too, right? What you learn in Unit 1 will be foundational knowledge also for Unit 2. So it is very, very important that you understand each unit before we move on to the next, or you will get what we call a snowballing effect, right? If you haven't caught up from Unit 1, Unit 2 is going to be hard to catch up on. So, a little bio review. What is the scientific method? Well, it's a step-by-step progress used by scientists to investigate questions, gather evidence, and draw conclusions based on experiments and observations. So the first thing you have to do is make an observation, right? And then from that observation, you have to ask a question. This part, you're going to explore the literature, right? And that's going to be really important because throughout this course, we're going to be writing formal lab reports. And in that formal lab report, you're going to have to require to bring background information so that you can base your understanding of the experiment on background information as well. So you need to be able to understand how to explore literature. From there, you will form a hypothesis, and we'll go into deep detail about how to do that, design an experiment, collect data, and draw a conclusion. How to collect data? Well, you have to understand what the expected versus actual results are and how to draw a conclusion from that. We will say we support the hypothesis, we reject the hypothesis, or we will form a new hypothesis and start all over again. So, how to make an observation. So, in this example, monarch butterflies can sometimes be found flying at night near very bright artificial sources of light, like streetlights. So, you're wandering around and you start to notice the monarch butterflies, which we are in a migration pattern for those. You start to notice that, hey, they kind of start gathering around these streetlights. So then you might ask yourself, what's causing this phenomenon? Can this be manipulated and turned into a repeatable experiment? So you start to explore some literature. You say, well, has anybody ever really looked at this? Monarch butterflies are diurnal, which means they are active flying during the day, resting, not flying at night, just like you and I. We don't fly, but we are active during the day and we rest at night. They have circadian rhythms, which are internal clocks that track day and night, just like you and I. We know our body knows when to release hormones because we have internal clocks that tell us it's day or nighttime. They are migratory, so they use sunlight as a compass and antennae, which helps track the sun during flight. And the proteins important for flight are processed at night while the butterfly rests. So, after you've read all the literature, these are the things that you've decided are really, really important to your observation. So, the scientific questions that the researchers might ask are, how does exposure to artificial light at night affect the number of monarch butterflies that initiate this flight? Then you form a hypothesis. A hypothesis is a testable explanation for an observation. It explains the relationship between variables and what the researcher expects to happen to one variable if another variable changes. You may have previously learned this structure for hypothesis, an if dot 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 then dot 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 because, but it does not need to be in this format. The if is the manipulative variable, right? So this is what the independent variable is. It's what we're changing throughout the experiment. The then is the responding variable. So this is the dependent variable. It's what we measured. It's what responded to what's being changed. The because is an optional explanation. Results either support or do not support / refute the hypothesis. Never, ever, 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 ever say that the hypothesis is correct. That is wrong. We are testing things. We cannot say with absolute that a hypothesis is correct. We can only say that our data or our results supports the hypothesis. So I want you to think pair share. So let's go back to the monarch butterfly example. Scientific questions begin investigated. How does exposure to artificial light at night affect the number of monarch butterflies that initiate flight? What is the possible hypothesis for this question? So you gotta ask yourself, well, what's the independent variable here? What are we changing? And what's the dependent? What are we measuring in our experiment? So exposure to artificial light at night increases the number of monarch butterflies that initiate flight. So if you break down this hypothesis, we would say the artificial light, the exposure of or not to artificial light, is the independent variable. That's what we are changing as scientists. Are they exposed or are they not exposed? And then we're going to measure the number of monarch butterflies that initiate flight when exposed to artificial light at night. So, in order to test your hypothesis, statistical test procedures to test hypothesis about a population based on samples. So we will perform statistical tests in order to test our hypothesis. It helps to determine if the observed differences between groups are statistically significant or are they due to chance. Why do we need math? Maybe perhaps some of you are listening to this video, you pause, your mouth is just agape because you are shocked that biology involves math. Of course biology involves math. We're humans. We are biased. We have to have a way that says this is happening. There is a statistical evidence that says that this is different. Because if not, we would just say with our own two eyes, yeah, that feels different. So yeah, we're going to do some math in this class. It involves two types of hypothesis. You have to have a null hypothesis. You can also call this the H naught or the H0. It's a hypothesis that there is no effect or no difference between the two groups of data. Observations are the result of chance. The alternative hypothesis, you will number them if you have more than one, but you can just call it HA or H1. If you have more than one, you would say H2, and so on and so on. Hypothesis that there is a relationship / effect difference between the two groups of data. Observations are due to a non-random cause, meaning that the differences that we see are due to what we are exploring, not just something that was random. That may be a new term for what you're already familiar with. This is what the researchers are claiming or studying. So in this class, we will have two hypotheses. We will have an alternative hypothesis and a null hypothesis. Anytime I tell you to state the hypothesis, I need you to state both, especially if there are stats involved, but we are going to make a habit and we are going to state both in this class. Why do we need a null hypothesis for testing? It provides a baseline, which we can use to compare our sample's results to. Allows for a hypothesis to be tested in a meaningful way using statistical tests. So if you are doing math, you must include a null hypothesis when you're doing research, but we're going to make a habit out of this class. We're just going to include both. Use the data to determine if there is a strong enough evidence to reject the null hypothesis. So when there is a null, you either support it or you reject it, meaning the alternative, if you reject it, that means the alternative hypothesis is supported by data. We're going to really go into stats more in Unit 5, but stats is super important for this class and for the AP Bio exam. So you really need to be able to understand what, when you're looking at data, what the stats is telling you. So let's practice. Determine a suitable null hypothesis for the monarch butterfly experiment. So go ahead, think of something, and then just jot it down. All right, so maybe you said, how does exposure to artificial light affect the number of monarch butterflies that initiate flight? So that is the alternative. The null to that would be exposure to artificial light has no effect on the number of monarch butterflies that initiate flight. So here you see in the very beginning, when we were introducing the monarch butterflies, we had a directional hypothesis. We said it would increase the number of monarch butterflies. Now that we have a null, we can just say it has no effect on the number. So here I want you to work on practice problems 2 and 4, or 2 through 4. So, does ivy grow better in areas with more sun versus in shady areas? So, this is our question. So, now we need to formulate one null and one alternative. So, our manipulated variable or our independent variable is there sun or is there shady areas? And then our dependent variable here is the ivy growing better, right? So does it increase growth or does it decrease growth? So you need to make a hypothesis based on knowing those variables. So the null here would be sun has no effect on the growth of ivy, or you could have said there is no difference in the growth of ivy between areas with more sun and less sun. I like the first one. It's way less wordy. And when you get to the AP exam, and if it asks you to state a null, the less wordy you are, the happier your graders are. An H alternative or an H1 could be areas with more sun exposure have increased ivy growth, right? Or we could have said areas with less sun exposure or shady areas have an increased ivy growth. Now, number 3, a group of students are interested in examining the potential effects of caffeine from soda on heart rate. One group of students hypothesized that soda consumption will not have an effect on heart rate, while another group proposed an alternative hypothesis that soda consumption will increase heart rate. What would be expected if the alternative hypothesis was supported? So, here we have the first part of this paragraph, right? When we're looking at hypothesis, it will not have an effect. So we know that's a null. And then the alternative said that the soda consumption will have increased heart rate. So the more soda you drink, the higher your heart rate, right? Heart rate increases after soda consumption. Number 4, a researcher suggests that small doses of hyaluronic acid, HA, will promote the uptake of water in human skin, thus reducing symptoms of dryness and dehydration, right? We might see a lot of hyaluronic acids in our skincare products. He suggests that HA binds to water and prevents it from evaporating on the skin. The researcher creates a skin cream formulated with hyaluronic acid and compares the skin of 100 people before and after the use of the cream. Formulate one null and one alternative hypothesis. Describe the data you would expect to see that would support the null hypothesis. So here we know that he's manipulating if there is hyaluronic acid in his product, right? And he's saying that I'm going to measure the dryness and dehydration of these people's skin. So a null here would be that adding hyaluronic acid makes no difference to the dryness or dehydration of a person's skin, right? Somewhere along those lines are what you have to write. An alternative could be the addition of hyaluronic acid will increase the hydration of somebody's skin, or it will decrease the dryness and dehydration of someone's skin. So here it is in writing, hyaluronic acid has no effect on skin dryness, or there's no difference in skin hydration levels before or after the use of hyaluronic acid. One alternative hypothesis is hyaluronic acid will decrease the symptoms of dryness, dehydration when used on skin. Alternatively, you could also say it could improve hydration, right? The most important thing I want you to take away from this is that you really, in order to form a hypothesis, you really need to understand from the question, what are the researchers changing? That's what we're going to call our independent variable. What are the researchers measuring, right? What's responding to the change? That's going to be called our dependent variable. And if you really can identify those two, you can form a hypothesis very, very easy. If the hypothesis was supported, I would expect that the see no significant difference in the hydration levels of the skin before or after the use of HA. Notice that they said no significant difference, right? That's the important thing about the null hypothesis. We need to be able to see that there's no significant difference, and we do that with using stats. Obviously, we didn't do that. This is a hypothetical problem, but we would have used stats to say that there's no significant difference. Now, how to do experimental design. Hopefully your eyes are not glazing over from this PowerPoint, but this is really important because 1, 2, and 3 of the FRQs in the AP exam come May really need you to understand what researchers are doing in experiments. They truly do pull research papers and pull questions from those research papers to see if you understand them. So hopefully you can kind of understand what's going on. So something very important that I notice in students a lot is that they don't understand the difference between control groups and control variables. So we're going to kind of try to nail that down today. So groups, we've got control groups and experimental groups. Very important that you understand the two difference. Control groups are used for comparison. You can also say to make a baseline. So it's you use them to make a baseline in order to compare them to the experimental group. It's helped to validate statistical analysis and increase confidence in the conclusion drawn from the experimental results. So they're needed to be there to show that there's some type of statistical difference in the control, or statistical same depending on the experiment, in the control groups versus the experimental groups. The experimental group receives the experimental treatment, which is our independent variable, being tested to observe its impact on the outcome, or your dependent variable. At least 3 trials are needed per group. Now, within the controls, there's 2 different ones. There's a negative control. It's a group not exposed to any treatment or exposed to a treatment known to have no effect. So if we're talking about bacteria, which is what I studied, that's what my degree is in, it's in microbiology, we often use no antibiotics as a negative control when we're determining if a bacteria is susceptible to a antibiotic. Water is a really good one as well because water tends to have no effect. Or you could just say the word buffer as well. Helps to ensure there is no effect when there should be no effect, right? If If I want to know if my bacteria is going to become susceptible to an antibiotic, I'm first going to put that bacteria on a media that I know it can grow in with no antibiotic. That way I know the bacteria can actually grow. Positive group not exposed to the independent burial, but is exposed to a treatment known to have an expected effect. So here, a positive control, if I'm thinking about a bacteria, if I want to know if a bacteria is resistant to or susceptible to a new antibiotic on the market, right? I'm working in pharmaceuticals. I'm first going to test antibiotic that I know the bacteria is susceptible to. That way I have some type of baseline to compare it to. So if this antibiotic worked for sure, I can now test my new antibiotic and use stats to determine was there a true effect on the bacteria with the new antibiotic. Ensures the experimental setup can produce a known effect, provides a reference point for what a known effect looks like as well. Some examples of negative controls. A researcher wants to test the effect of caffeine on heart rate. Researcher will give negative control group a treatment that is known to have no effect on heart rate. And it's going to be water because water should not affect your heart rate. Water is known to have no effect on heart rate with consumption. If the water affects the heart rate in a negative control group, then there must be another variable affecting the heart rate, or the water is contaminated. Some examples of positive controls. A researcher wants to test the effect of a new antibiotic on a strain of bacteria, right? So this is what I was just saying previously. How would the researcher know the new antibiotic experimental group is actually effective? It'll use an established antibiotic that is known to work. We'll call this the positive control group. If the experimental group fails, but the positive control is successful, it is likely that the tested antibiotics are ineffective. All right, I'm going to stop the video here because I'm running out of recording time. Please pick back up on the new video next.